Hi, my name is Andrew. This is my project. Um, when we talk about integration, um, especially at the animal level, we want to look at how animals integrate diverse sets of information. Um, this can be internal information or external information, but we want to see how when we're presented with two different sources of information, how we combine them. At the cellular level, there's two different types of summation of neuronal inputs that we're interested in. On the right, we have an example of linear summation or integration, where the two inputs are the output that is formed from the two inputs is the sum of the two inputs. Whereas on the left, we have superlinear summation, where the um, combined the combined response to the inputs is greater than the linear sum. We want to study whether this happens in a living organism because those results come from brain slices. So we're going to use C. elegans to study this phenomenon, which has 302 neurons. Its connectome is fully mapped, and its body is transparent so we can visualize neuron activity. We're going to use this candidate neuron titled AIB, which we chose as a candidate because it has a lot of clustering of synaptic inputs. And two of the sensory inputs onto this neuron, AWC and AIA, are known to be responsive to sensory information, and we're able to activate these neurons without activating a bunch of other neurons around them. We're going to use this experiment where we provide odor to stimulate AWC, and we provide light to stimulate AIA, and we look at how providing both of these sensory inputs at the same time affects AIB compared to providing the inputs individually. We use a tool called channel rhodopsin in AIA, which causes the neuron to depolarize when it's exposed to light. At the same time, we're going to be monitoring activity of the neurons using a genetically encoded calcium sensor, which fluoresces when calcium ions are bound to the protein. This is what the neuron activity looks like under the microscope. They're flickering in response to changes in the potential. On the right here are traces of what it looks like when the fluorescence changes. I want to model AIB using these two equations. The top one is just the sum of the neurons involved in AIB's activity. AVA is a motor neuron that is very correlated with AIB's activity. On the bottom, we have an equation that involves this nonlinear term AIA times AWC. As you can see in the little table below, AIA is only providing information when both AIA and AWC are providing information. These models are useful because we can see that they fit the data when we try to predict an AIB trace from them. And when we evaluate the fit quality, we see that the model with the nonlinear term and the linear terms has less variance in its residuals across all experiments. This tells us that the model is more consistent in how it fits the data. I'd like to conclude with some future directions. We want to collect from more worms. This data is only from 9 worms. And we want to also assess the fit quality to make sure that the nonlinear term is actually useful. And we want to make sure that providing odor or light is not stimulating both neurons. I'd like to thank Amanda Ray, the PhD student on this project, and Dr. Andrew Gordis, my supervisor. Thanks.